My name is Eliza White, and it is March 30th, 2026. Cells produce many different types of macromolecules that follow different intracellular pathways depending on their function. The pathways for the steroid hormone cortisol, which is a lipid, the protein hormone glucagon, which is an extracellular protein, and the cytoskeletal protein actin illustrate how molecules are produced and transported inside a cell. The synthesis of the steroid hormone cortisol begins in the smooth ER of the adrenal cortex cells. Unlike proteins, steroid hormones are derived from cortisol rather than synthesized on a ribosome. Cholesterol is transported into the smooth ER, where a series of enzymatic reactions convert it into cortisol. Because cortisol is lipid soluble, it does not need transport vesicles or secretory vesicles for export. Instead, once synthesized, it diffuses through the cell membrane and directly enters the bloodstream, where it can travel to target tissues and regulate metabolism and stress responses. The protein hormone glucagon follows the classic secretory pathway of the endomembrane system. Its production begins on a ribosome attached to the rough ER. As the ribosome translates the glucagon precursor protein, it enters the lumen of the rough endoplasmic reticulum, where it begins folding and undergoes initial modifications. The protein is then packaged into transport vesicles from the Golgi apparatus. In the Golgi, glucagon is further modified, sorted, and packaged into secretory vesicles. These vesicles travel through the cytoplasm and fuse with the cell membrane, releasing glucagon outside the cell by exocytosis, so it can act as a hormone that raises blood glucose levels. Finally, the cytoskeletal protein actin is used inside the cell and therefore follows a different route. Actin synthesis begins on a free ribosome in the cytoplasm and is rather than on the rough endoplasmic reticulum. Because actin functions within the cell, it does not enter the Golgi apparatus or the vesicular transport system. Instead, once translated, it folds onto its functional structure and polymerizes into filaments that form parts of the cytoskeleton. These actin filaments provide structural support, help maintain cell shape, and enable processes such as cell movement and intracellular transport. Together, these three pathways demonstrate how cells use different organelles, including the ribosome, rough endoplasmic reticulum, smooth endoplasmic reticulum, Golgi apparatus, transport vesicles, secretory vesicles, and cell membrane to produce and distribute macromolecules depending on their final destination.